Hi team, welcome to my video about RNA processing. And we're going to start with this question. A geneticist isolates a gene for a specific trait under study. She also isolates the corresponding mRNA. Upon comparison, the mRNA is found to contain 1,000 fewer bases than the DNA sequence. Did the geneticist isolate the wrong DNA? So our options are: yes, she isolated the wrong mRNA because mRNA is made from a DNA template, so it should be the same length as the gene sequence. Well, we know that mRNA is made from a DNA template, but this is the question: should it be the same length as the gene sequence? Our next option is yes, she isolated the wrong DNA because the mRNA should contain more bases than the DNA sequence. Well, that's incorrect, so we can just cross off number 2. Number 3, the option is no, she did not isolate the wrong DNA. Instead, the final mRNA contains only a subset of the base pairs of the gene. So this is definitely a possibility. And then number 4, no, she did not isolate the wrong DNA. Instead, the mRNA is shorter because it was partially degraded after it was transcribed. Well, the final conclusion from this question should be that number 3 is correct. The final mRNA contains only a subset, that is, only the exons are included while the introns are removed. It wouldn't be appropriate to say that the mRNA was degraded. We're going to talk about how the mRNA is shortened on purpose, whereas degraded sort of sounds like it's being thrown away. So the mRNA is shortened on purpose, yes, but degraded has a really negative connotation. So I would say 4 is incorrect. And then up here, we're going to talk about in eukaryotes, no, the mRNA is not the same length as the gene sequence, but in prokaryotes, yes, it would be true that the mRNA is the same length as the gene sequence. So let's get into some of the details of this. What we'll be describing in this video is RNA processing. So that is how we go from the transcribed mRNA into the mature mRNA that is released into the cytoplasm of the eukaryotic cell so that it can be translated. And all of these RNA processing steps that we'll talk about, capping, splicing, adding the poly A tail, so here's the cap and the poly A's, and you can see that these introns have been removed. Okay, so that'll be splicing. All of these things only happen in eukaryotes, and that has to do with the presence of this nuclear membrane. Because we're transporting the RNA from inside the nucleus out into the cytoplasm, we need to have some quality control steps. And many of these processing steps are quality control. In prokaryotes, on the other hand, because there's no nucleus, translation can begin as soon as mRNA transcription occurs. There's no separation of transcription and translation. So I'm going to write of transcription and translation. There's no time for any of these RNA processing things to occur, but there's also no need for them to occur. So let's talk about the details in eukaryotes of RNA splicing, capping, and polyadenylation. Our learning goals today for this set of slides is going to include talking about collinearity, which is true in prokaryotes and less true in eukaryotes. We're going to talk about the processing steps of RNA, and we're going to talk finally about alternative splicing. Let's start by thinking about collinearity. Now, in the olden days of science, scientists took DNA, which of course is double-stranded, and RNA, like mRNA, which is single-stranded, and heated the DNA and RNA so that all of it was single-stranded, and then cooled this material back together so we could see where the sequences of RNA matched up with the sequences of DNA. I think you can see here when RNA and DNA hybridize, we sometimes end up with a loop of material that's not part of the hybridization. And that's what's shown in this micrograph over here. We've got some linear regions, over here. But then we've also got some loops that are not included in the linear region. What are these loops? When a eukaryotic mRNA is hybridized to the complementary region of DNA, loops of unhybridized DNA are seen. These loops, well, do these loops correspond to the non-coding regions of the gene? Are these loops not transcribed into mRNA? Do these loops demonstrate that genes and proteins are not collinear? Option D is both A and C are true, and option E is A, B, and C are true. Well, let's talk about this. The non-coding regions of the gene, non-coding refers to things that are not turned into protein. Okay, so examples of that could include introns. So these loops could include introns. Now, introns are transcribed into mRNA. Introns are transcribed, but then they are removed. So we know that B can't be true. The loops do demonstrate that genes and proteins are not collinear, because if the genes and the mRNA were both, so let's write DNA and mRNA, this would be an example of a linear relationship. You can imagine also that if we had a number of amino acids in a protein and number of bases in an mRNA, if this were the relationship we saw, we'd expect this to occur. But because we have some loops, so we saw loops, okay, that will demonstrate that the genes and the proteins coded by mRNA are not collinear. Okay? So actually both A and C are correct here. Let's talk more about the details of this. Here's a diagram that shows the idea of collinearity. When we have a DNA sequence up here at the top, which is transcribed into mRNA, the number of bases in the DNA and the number of bases in the mRNA are the same, indicating that these two are collinear. They're linear together. And then when we group the bases in codons, or sets of 3, triplets, each codon corresponds to a single amino acid. So even though there's a 3-to-1 ratio, the mRNA and the protein are still collinear. Now, based on studying prokaryotes, we know this is true. mRNA, so DNA, mRNA, and proteins are all collinear. But the previous experiment where we hybridized DNA to RNA in eukaryotes, DNA and mRNA were shown to have loops. They're not collinear. So what is going on with eukaryotes? Let's start by thinking about the structure of a mature messenger RNA, or mRNA, in eukaryotes. So what we're going to be talking about for the rest of today will be true for eukaryotes, as these are the ones with the non-collinear RNA and DNA. Our mature messenger RNA has a protein coding region with an AUG, and a UAA, or UAG, or UGA for a stop codon. And this is often something that you already know to look out for, the start and stop codon. But in fact, the mature mRNA can have regions that are upstream and downstream of the start and stop codons. Because this start refers to the start of translation, and the stop refers to the stop of translation. Recall that transcription would have started further back here. Transcription would start, and then transcription will end. This is our terminator region. Okay, so these regions between the start of translation and the start of transcription are referred to as the untranslated regions, or sometimes the UTRs. So the 5' untranslated region is upstream. The 3' untranslated region is downstream. And then the center of this mRNA transcript is the protein coding region that will get turned into amino acids. The last thing I'd like to point out is this yellow region here. This is referred to as the Shine-Dalgarno or Kozak sequence. This is a consensus sequence. I'm just going to draw it down here. This is a consensus sequence. And as we'll talk more about during the process of translation, this is where the ribosome binds to start translation. So a sequence like this would be present in both eukaryotes and prokaryotes, and in eukaryotes it's called the Kozak sequence. So we've got our 5' untranslated region, we've got our ribosome binding site, we've got our protein coding region, and then we've got our untranslated region at the end. Let's talk more about the 3 processing steps that occur to help us make this mature mRNA. The first step in processing the pre-mRNA is to add a cap to the 5' most end of the mRNA. So here is the last base of the mRNA, whatever it might be. But on the 5' end of that base, we're going to add this 7-methylguanine cap. Shown to the left here, you can see this is a G base upside down. Maybe an easier way to write that is a G upside down. So we've got 1, 2, 3 phosphate groups, deoxyribose sugar here, and then we've got our G purine over here on the far left. And that's what's attached to the 5' most base of the mRNA. And this capping process is a quality control step. If the cell sees an mRNA that has a cap, it tells the cell everything is okay. This mRNA is good to go. We can continue processing it and treating it like a friend. No need to get rid of it. Let's look at the capping enzyme complex. You can see here that we've got RNA polymerase shown in the center here, with the DNA running through the RNA polymerase, and then the mRNA coming out the other end of the polymerase. And attached to the polymerase is this 5' cap synthesizing complex. So as soon as the mRNA reaches that cap synthesizing complex, this 5' cap is put on, and the RNA will continue processing as RNA polymerase moves along the DNA, and ultimately the mRNA, when it's released from the RNA polymerase, will have this cap attached to it. Now that we've capped the mRNA, I need to draw the 7-methylguanine cap right there. Now that we've capped the mRNA, the next part of RNA processing is cleavage and polyadenylation. And remember, these are steps that are only going to occur in eukaryotes. We've got, we're showing here transcription with the help of RNA polymerase. And as we discussed, let's say that right here is the gene that we're interested in transcribing. But if we look at this RNA, you can see that RNA polymerase has transcribed a lot of extra material down here that we don't want to ultimately turn into protein. And so a next step that we'll have to take is to cleave or cut off a portion of the mRNA that we don't need anymore. So here we can see a consensus sequence. This is where a nuclease enzyme will bind. So it'll bind and recognize down here. And then over here is where it will cut. Okay, so we've got the pre-mRNA that's already been capped, and now the cleavage enzyme will look for a consensus sequence to line it up and cut or cleave the mRNA. Once the mRNA has been cleaved, I have to redraw on our 7-methylguanine cap. Once the mRNA has been cleaved, it's ready for the next step, that is polyadenylation. So there'll be another enzyme here that will add a poly-A tail, which you can see is just a chain of A bases, adenines. So we're adding a poly-A tail, and just like this 5' cap, this poly-A tail is a quality control symbol. The longer the tail, the longer the mRNA will be able to stick around inside the cell. So we could draw a little graph that says length of the poly-A tail and then the lifetime of the mRNA. And they're pretty well related. A short tail means this mRNA is going to be degraded quickly. A long tail means this mRNA can stick around and be translated for a long time. The third step to processing RNA in eukaryotes is the removal of introns. So let me show you what this means. In the blue line here, we have our chromosome DNA, and this represents a gene called G-alpha-49b. G-alpha-49b. And right below it, we've got the transcript, that is the mRNA. And you can see here that there are some fat bars and some thin bars. The fat bars represent the exons of G-alpha-49b. And then the thin bars represent the introns. These are all little introns. Now, when transcription occurs originally, the pre-mRNA will look just like the gene. But these intron regions, so I'll label this the pre-mRNA. Now, of course, it'll get capped, it'll get cleaved, and then a poly-A tail will be added onto it. And next, we need to remove these regions that are introns, so that all that's left in the final mRNA are the exons, and they'll be butted up next to one another with no space left over for the introns. Our mature mRNA with its poly-A tail will have only exons, and the introns will be removed. What does that process look like? The first thing I want you to know about is if we zoom in to an intron. So you can see here, I've got an exon on the right, we've got an exon on the left, and we're looking at the sequence of the pre-mRNA. Now here in yellow in the middle is the potential sequence for an intron. And what we're looking at underneath the intron is the frequency of these base sequences occurring. You can see at the end of an exon, at the beginning of an intron, it's 100% likely that we're going to start with a GU. And then at the end of the intron, it's 100% likely we're going to end with an AG before we go into this next exon. Okay. Now it's pretty likely up here that we have an A or a G before the intron. But that's only pretty likely, right? It's pretty likely that our second exon starts with a G. But of all of the thousands of introns that were sampled here, 100% of them started with a GU and ended with an AG. Okay. And there's one more important sequence in the middle of the intron. It's an A, and here it's labeled the branch point A. In order to remove these introns, we need the help of an enzyme complex called the spliceosome. The spliceosome will look for these intron sequences as consensus sequences. So this GU, A, and AG are the consensus sequences for the spliceosome. So let's talk about what the spliceosome does once it recognizes these regions of the intron. Here's a zoomed in version of what's happening during the spliceosome steps of splicing. Okay, so we've got our exon 1, our exon 2, and here in yellow is still our intron. The first thing that happens is this branch point A sort of attacks the end of exon 1. And the result of that is that we make a little loop here. All of the intron is now in a sort of loop, but the end of exon 1 is free. It's not attached to anything. So next it will attack the beginning of exon 2. And ultimately, exon 1 and exon 2 are spliced together. That is, they're connected by a phosphodiester bond, shown right here. And the intron, in the form of a loop or a lariat, has been removed. Now, this is a really molecular organic chemistry sort of summarization of what's happening during splicing. So let's look at another version. This is a second way of looking at splicing. And you can see again, we've got our exon 1, it ends with an AG, and the intron starts with a GU. Our exon 2, right before it in the intron, is an AG. So we've got those consensus sequences there. And then here I'm showing you some of the spliceosome proteins. Remember I said the spliceosome is a protein complex, so that means there's many proteins working together. And these two proteins, U1 and U2, are important for recognizing two of the consensus sequences in the introns. With the help of these other spliceosome proteins, U1 and U2 are brought together. Ultimately, U5 and U2 help bring together the branch point A with the front part of the intron. And then the first exon is allowed to connect together with the second exon. Now we've got exon 1, exon 2 together, and the loop of intron sequence, the loop or the lariat of intron sequence. So we've got these spliceosome proteins helping to bring together this loop of the intron so that exon 1 and exon 2 can come together and be joined together. Last but not least, remember this is only happening in eukaryotes. Prokaryotes don't have introns, so they don't need to splice them out. That means they also don't have a spliceosome. So we've talked about the idea of splicing of pre-mRNA into mature mRNA. But what's the deal with alternative splicing? So let's just look at the left side of this diagram here. We're starting with our DNA. It's transcribed into a pre-mRNA. And then the mRNA is spliced, removing all of these 4 introns into a mature mRNA with 1, 2, 3, 4, 5 exons, and then that's translated into a protein. Okay, and for many, many years, scientists believed that this left side of the diagram was the only thing that occurred. But now we know that this version of RNA splicing is not the only version that's possible. In the same person's body, we can do this secondary splicing, or let's call it alternative splicing, where here we're splicing out, we're keeping exon 1, 2, you can see we're splicing out intron 2 and 3 and also exon 3, but then exon 4 and exon 5 are left in. So now we've got a different version of a mature mRNA that's going to code for a slightly different or alternative protein. And this protein might have a different structure and a different function inside the cell. In this example, we're looking at something that might be changed during a disease state, where healthy people tend to have protein A, but in the presence of disease, protein B is more likely to be occurring because this alternative splicing form is what's occurring during splicing. So let's look at an example of that. Here's an example of a gel where we're looking at mRNA levels. And on the left-hand side, you can see we've got MW, that stands for molecular weight ladder. That's why we've got regular intervals here. It's not important necessarily what those size markers are, but it is important to remember that a gel can separate DNA and RNA based on size. Okay. And then we're looking at the mRNA from the gene LEAP2. And we're looking at it from 6 different tissues: liver, kidney, colon, lung, trachea, and heart. If we look at the 3 tissues on the left-hand side here, you can see we have 1, 2, 3, 4, 5 different sizes of mRNA, but the most abundant one is this one that's about 350 base pairs long. So the mRNA that's most common in liver, kidney, and colon is 350 base pairs long. So the LEAP2 protein is going to be translated from the LEAP2 mRNA. And in the liver, kidney, and colon, we have one that's going to have about 350 base pairs in mRNA. That'll mean the protein is a little bit bigger than 100 amino acids. Now, for comparison, in the lung, trachea, and heart, the 350 base pair mRNA is still present. So we're still making that 100 base pair protein, but the more abundant mRNA is this one at 550 base pairs. That means this mRNA is going to be making a protein of closer to 200 amino acids, which could have a similar but totally different function. So these are all the possible alternative splice variants for LEAP2 that occur in our very different tissues that have maybe slightly different jobs for the different shaped proteins. All right, so that's RNA processing. We'll be back to talk more about translation of proteins from those RNAs soon.